JP sir, am I audible?
Yes, sir.
Parasuraman sir.
And Dr. Saravanan, we can start.
Preeti madam, can start madam.
A very good morning to one and all. On behalf of MNR College of Pharmacy, I wholeheartedly welcome all the delegates to the webinar on applications of molecular modeling in drug discovery. MNR College of Pharmacy has been organizing webinars, e-conferences, student development programs on the online platform since the onset of the pandemic in 2020. We have more than 1,000 participants registered to every webinar, which is an indicator of the quality of webinars. The sessions were streamed live on YouTube to reach out to a large number of students and professionals. The appreciation and positive comments we received from Zoom and YouTube platforms have encouraged us to continue disseminating information and knowledge through the webinars to a large number of students and pharma professionals in order to support them in their skill development during this pandemic. Science of today is the technology of tomorrow. The topic of the day, applications of molecular modeling in drug discovery, was chosen on the context of advances in the field of molecular modeling. It has become a valuable and essential tool to medicinal chemists in the drug design process and describes the generation, manipulation, and representation of three-dimensional structures of molecules and associated physiochemical properties. Today, we have with us subject expert Dr. P. Parasuraman, who heartfully consented to elucidate to us the various possibilities of molecular modeling as a powerful tool for drug design and molecular docking. Now I invite Professor Dr. G. Saravanan to introduce the speaker and MNR College of Pharmacy to us.
Thank you, madam. A very good morning to one and all. It's my great pleasure to introduce MNR College of Pharmacy to the gatherings. MNR College of Pharmacy is one among the top 73 grade institutions run by MNR Educational Trust. MNR Educational Trust was established in the year 1974 by our chairman, Sir Sri M. N. Raju Garu, with the noble aim to shape the students into proud citizens of India. Our vice chairman, Sri Ravi Verma Mantena, adds a perfect blend of modern approaches to achieve the goal. MNR College of Pharmacy is established in the year 2004 and it is approved by Pharmacy Council of India and All India Council for Technical Education, New Delhi. MNR College of Pharmacy is affiliated to Osmania University, Hyderabad, and we are offering the courses B Pharmacy, PharmD, M.Pharm, and PharmD PB. Recently, MNR College of Pharmacy is approved as a research center by Osmania University, Hyderabad. State-of-art pharmaceutical laboratories is present in MNR College of Pharmacy, more than 10,000 volumes of specialized books, and we have a subscriptions for journals and digital library facilities is available for both students and staffs. Along with curricular activities, we are also encouraging our staffs and students to participate in various co-curricular activities and extracurricular activities. More than 50 faculty members are working in MNR College of Pharmacy. Among them, 20 faculties are with PhD. The student team and the staff team is led by our distinguished principal, Dr. V. Alagarsamy sir, who is working in MNR College of Pharmacy since 2007. Sir is having 23 years of teaching and research experience. Sir has 150 plus research paper for his credit, and Sir is an editor of Anti-Infective Agents, a well-known Bentham Science journal. Sir is authored 6 popular books in the pharmaceutical chemistry field. Recently, Sir is authored a textbook of organic chemistry. Sir is a recognized PhD guide in many Indian universities. Sir has been ranked among top 2% scientists in the world by Stanford University, USA. He is the only one ranked in both Telangana and Andhra Pradesh, and Sir is placed at 22nd place in the subject of pharmacy. So, in MNR College of Pharmacy, we taken a various special initiatives for students to achieve a university rank and placements. Our chairman sir always believes that education and disciplines are the two eyes. So hence in MNR College of Pharmacy, we are providing the education with the disciplines, and we are providing a favorable environment for studies. With this brief introduction about MNR College of Pharmacy, it's my great pleasure and honor to introduce my friend and my research collaborator, Dr. Parasuraman P, to these gatherings. Dr. Parasuraman completed his B Pharmacy in 2009 in EGSP College of Pharmacy, Nagapattinam. Later in 2012, Sir completed his postgraduate diploma in clinical research and data management in Bharatiyar University, Coimbatore. In 2000, completed his M Pharmacy in the specialization of pharmaceutical chemistry in KMCH College of Pharmacy, Coimbatore. In 2016, Sir got his PhD from Annamalai University, Chidambaram. The area of research interest of Parasuraman P is drug design, lead optimization, QSCR, molecular modeling, ADME modeling, structural based drug design, ligand based drug design, homology modeling, pharmacophore analysis, and analog design. Sir is a recognized PhD guide of MS Ramaya University of Applied Sciences, Bengaluru. Sir is a registered pharmacist in India, and he is a life member of APTI. Sir is a editorial board member and peer reviewer of many reputed journals and publishers. Sir is a approved examiner and question paper setter for many reputed universities in India. Dr. Parasuraman having 4 patents for his credit, and till date he has an 3 book chapter for his credit. He published his research findings in 43 international and national journals with a good impact factor. Sir is presented his research in many international and national conferences by oral as well as in a poster mode. Sir attended 6 workshops of these molecular modelings, and Sir delivered many guest lectures in various universities all over India. Sir acted as a chairperson and subject experts for many national and national conferences. Till date, Sir guided 1 PhD students, 4 M Pharmacy students, and 4 B Pharmacy students for completing their degree. Sir have many awards and honors for his credit. In 2012, he got an UGC Basic Research Fellowship. Under this fellowship, Sir completed his PhD. And in 2014, Sir got a Genix Bio Young Scientist Travel Grant Research Award. In 2019, Dr. Parasuraman received Best Researcher Award and Top Performing Mentor Award. Sir started his career after his M Pharmacy as a full-time research scholar in Annamalai University in 2012. And after completion of his PhD in 2015, Sir started his career as an assistant professor in Noori College of Pharmacy, Andersonpet, Karnataka. Later, Sir worked as an assistant professor in Hillside College of Pharmacy and Research Center, Bangalore from 2016 to 2017. From 2017 onwards, Dr. Parasuraman P is associated with MS Ramaya University of Applied Sciences. Currently, Sir is working as an assistant professor in the Department of Pharmaceutical Chemistry, Faculty of Pharmacy, MS Ramaya University of Applied Sciences, Bengaluru, Karnataka. With this brief introduction, now I am requesting Dr. Parasuraman P to deliver his valuable lecture to these gatherings. Sir, please, sir, now the stage is yours.
Thank you, sir. Thank you so much for the brief introduction. And moreover, good morning to one and all. Thanks for the opportunity given by the MNR group of institutions. So I'll share my slides. Slides are clear, sir. Shall I start?
Yes, sir.
That's fine. So a very good morning, one and all. The topic which I'm going to discuss today is applications of molecular modeling in the field of drug discovery. So the term we are discussing about the word drug and all. So what is the drug? First, we should know what is the drug. The term drug means any particles which is intent for the use in the either the diagnosis or curing or mitigation or treatment or prevention of disease in a man or other animals. So a drug, it may be either a chemical, biological substance, or it may be a synthetic or non-synthetic substance. So this is about the general outline about a drug. So where we are getting the source, from which source we are getting the drug. So we are from the natural source as well as the synthetic source. Natural source in the sense from animals and plants. Animals, we are taking insulin from pig and cow, growth hormone, and from plants, digitalis morphine. These are the some examples where they are getting the sources. So n number of drugs, n number of moieties we are obtaining from animal source as well as the plant source. And then other than that, inorganic substances and synthetic. In synthetic, it will be further classified. Almost some 70 to 80% of the drugs are chemical only, chemical drugs only, synthesis only. And then from the biological one, that is penicillin, biotechnological, human insulin. So these are the some sources of the drug where we are obtaining this. So what is the need for a new drugs? As everyone are studying the biological classification or else the pharmacology classification or the medicinal chemistry classification, we can able to see in a particular class of a drug, we may have minimum of 5 to 6 drugs, an average of 5 to 6 drugs are there. But even though for the particular class we are finding a new drugs, what is the need? Why we need the drugs? What are the reasons? So the first reason is unmet medical need, because new diseases are arriving. As of now, we are suffering from COVID pandemic situation. Past 2 years we are suffering, but we couldn't able to find a proper medicine for that. And then the previous one is swine flu. And then due to low efficacy of the drug, so what are the drugs they have identified? They are not up to the mark to produce the expected pharmacological action. So that dementia and cancer, for cancer and all, the drug price will be high, those things and all. And then the side effects, if you take the antidepressant and antipsychotics, there are n number of side effects. N number of side effects are there for antidepressant drugs and antipsychotics. So we need a new drug. And then the cost of the therapy. If you take interleukins, Alzheimer's disease, spinal injury, so for such kind of diseases, the cost of the therapy for the treatment will be high. So as a researchers, what we have to do, we have to find a medicine which should be affordable by each and everyone. Either from the poor country or poorest country, they should afford for the proper treatment. So a new drug discovery is required. And to sustain the industrial activity, if you take the word called as patent, if a company is finding a drug for 15 years or 10 years, there will be a tenure. Within that time only, they can able to patent. Means that the patent will have an life period. So after that, anyone can able to manufacture. That is generic. If you take a branded drug and generic drug of a same molecule, the price will be of 10 times for the branded one and very low for the generic one. So to overcome, to maintain the peoples, to give the employer, so such kind of thing, they are finding out the new drugs. So and the industry employs thousands and makes a massive contribution overseas earnings. So to that, it is required. And then the most important thing is the drug resistance. The drug resistance in the sense, most probably antibiotic drug resistance and anti-cancer drug resistance. Antibiotic is the most common thing, because we people will never able to go for a proper treatment. That for if you want to take an antibiotic, it should be taken in a proper class. It should be a proper course should be taken. 3 days you have to be taken with a proper prescription, with a proper recommendation from the physician. But we people will directly go to the medical, will purchase, will get the medicine for 1 or 2 days, will take once the disease, I mean, once the fever goes, we'll leave that. But what happens, the microorganism which we are having, it will start to create the resistance. So once the resistance is created, we can't able to treat the same disease with the same drug, because the drug got adapted to the disease. So the microorganism will definitely up, so it will start to resist it. So for that kind of situation, we have to find out the new medicine. So these are the requirement why a new drug is required. So drug discovery and development in general, in olden days, how the drug discovery was this, how the discovery happened, drug discovery. First thing is they will identify the disease. Once they identify, they will isolate the protein involved in a disease. If we take any disease or disorder, there will be either the protein in the sense, the enzymes or receptor will play a major role. So we have to isolate that which is responsible culprit, which protein is responsible, which receptor is responsible. Until unless you find which is the culprit, we can't able to treat that particular disease. That is the thing. So to isolate the protein, it will take 2 to 5 years. Then what you will be doing, will be finding a drug effective against the disease protein. Then we'll go for a preclinical testing. Then we'll scale up the formulation and we'll file the IND, investigation of new drugs. So to trial with the humans, we'll be filing the IND. Then human clinical trials will take place for 2 to 10 years. Then we'll be filing the NDA and it will be marketed for the people's use. So these are the this is the platform how the drug discovery process will take place usually. So isolation of a protein, it will take 2 to 5 years. Finding a drug, 2 to 5 years. Preclinical testing, 1 to 3 years. And scale up, it will take some time. And then into clinical research, 2 to 10 years. So this is the road pathway where how the drug have been discovered. And then only it will be marketed. And FDA approval, it will take 2 to 3 years. So what are the complexity? What are the issues we are facing during the drug discovery process? So finding a drug molecule, it's not a simple task. So we should have a 10,000s of molecules. Example, 10,000 drug candidates with the valid biomedical hypothesis. If you are choosing a particular derivative or particular moiety, that it should have a proper hypothesis. With that hypothesis, if you are choosing 10,000 drug molecules, it has to satisfy these criteria. It should be a targeted one. It should be a selective in nature. It should be potent. It should be physically stable. It should be soluble. It should be permeable, metabolically stable. It should not induce anything. That is non-inducing agent. It should be a reversible one. It should be a durable, non-teratogenic, non-mutagenic. It should be a patentable, and it should be a manufacturable. So all these criteria, if a drug is fulfilling, out of 10,000 drug molecules with a valid medical hypothesis, it will reach to only one molecule. Only one drug molecule will be marketed. So these are the primary criteria. This is the complexity of a drug discovery process. So how the technology is impacting? What are the technologies we are using? The same process only. We have to identify, isolation, finding a drug. So these are because we can't able to avoid human trials. So that has to be done. But prior stages, we can able to use the applications of technologies, the technological development. So where it plays, genomics and proteomics plays a major role. Potentially, they will produce many more targets and personalized targets. So here, if you are carrying out a genomics and proteomics process of a particular disease, a particular thing, they can able to easily identify which is the culprit target, and which is the personalized target that can be identified using genomics and proteomics. Then high throughput screening. So if you have a 1 lakh of molecules in the lab, you can able to screen for a particular enzyme, and you can able to identify things. And the same way, if you're carrying out using the system, it will come under virtual high throughput screening. Using the computer to predict the activity. And then comes the combinatorial chemistry. So it will produce a vast number of compounds, different combination. Based on the combination, it will give out. Then comes the molecular modeling. It is based on the computer graphics models and help the improve in activity. Then as usual, it will enter into in vitro and in silico ADME models, which is a tissue and computer model begin to replace animal testing. So nowadays, in pharmacology, they are using the simulation software to find the activity, means using system without using animals. So drug discovery technologies. So drug discovery technologies, where how it is used for a target identification. Genomics will play a role, gene expression profiling, and proteomics. These three will play a role in finding the target, which is the culprit agent, which enzyme is responsible, which protein is responsible, which receptor is responsible for causing the disease. And then the next one comes, as we have identified, we have to validate it. That is target validation. Either we can able to validate the target by gene knockout or inhibition assay. So gene knockout in the sense nowadays the animal models are available, the knockout animals. So using that we can able to identify whether the particular target which we have identified using genomics, gene expression, and proteomics is right one or wrong. Is it a proper one or not? So to identify those things, it can be done. And then it comes a lead identification. Next one is lead identification. So we have found out the disease. We found out the disease condition and found out the protein, and we have validated the protein, or then we have to find out the proper drug. So lead identification. Lead identification either through high throughput screening. I have thousands of molecules. Out of thousands of molecules, 100 molecules belong to one category. Next 100 belong to another category. Like a quinoline derivative, stenolin derivative, like that 100 I have been doing, and from that I can able to opt out which type of nucleus is potent, and then based on that I can able to go further. And then fragment based screening, only the particular fragment which is active site, like mimicking the activity, which the active site of the protein can be mimicked using the fragments of a chemical structures, that can be done. And then lead optimization. So medicinal chemistry driven optimization, and then QSR model, ADME profiling, this can be done. So these are the four regions where the computational tools will play a major role. Then preclinical phase, that is pharmacodynamics, pharmacokinetics, ADME, and toxicity testing through animals, that we have to carry out. And then it comes to the human trials. This is how the drug discovery technologies plays. So we are discussing about the CAD. CAD is a computer drug design, or computational chemistry, computer drug design. This is whose major goals are to create an efficient mathematical approximation and computer program that calculate the properties of future drug molecules, and thus helping in the process of drug design and discovery. In simple term, what we can able to say, a computer drug design using the computers, like a programs with a mathematical calculation. Already I can give an example, Lipinski rule. We are saying the Lipinski rule of 5. So how he have made the rule of 5 in the sense, what are the drugs have been marketed, he have analyzed and he made, if a drug is having if the molecular weight is not more than 500, he have created a hypothesis, right? The same way here will be making the hypothesis, mathematical calculation can be made. So based on that, if I'm giving a model, if I'm giving a new designed one, this systems will say whether this molecule will work out or not, will have an efficacy or not, what its their absorption value, what it will be their physical chemical property. So for this, the process is called as computer-aided drug design process. It is a mathematical approximation. Just it's a calculation, mathematical theoretical calculation only. Like how we are saying with the nature, with the physical nature, like a human in the sense we can able to identify if there is a monkey and a human, we can able to say because we have that features. What are the features? Oh, so these are the features. It belongs to human category. It belongs to animal category. The same way systems we are going to train. We have trained. So what is the need for a CAD? What is the need for the CAD? Why we have to enter into the field? So drug discovery today are facing a serious challenge because of increased cost and enormous amount of time taken to discover a new drug, and also because of rigorous competition among the different pharmaceutical companies. So the primary thing is the increased cost. Yes, because drug discovery is $1 billion business. It's $1 billion business where they will invest in crores and crores and crores. So they have to get back. Many drugs will fail in their phase 3 also, phase 2 also, phase 1 also. Whatever they have invested, it is lost. It is lost. So the cost of the therapy, I mean, the cost for the investment for the discovery is more, to avoid that one thing. And second thing is enormous amount of time taken. So the time taken process to identify, as we have discussed in the previous slides, 2 to 5 years it will take, and then for the identification of drug 2 to 5 years, so it will take 10 years of time minimum. So to avoid those things, these two drug discovery process involved. And then the third comes the competition between the pharmaceutical companies to sustain in the market, because many companies are working for a monopoly product. Monopoly in the sense, what are the rate they are fixing, that will be the price. If 2 to 3 companies are selling the same product, they can't able to quote more price because of the marketing strategy. So many companies are involving in identifying a new drugs for as a monopoly product. So here we comes the molecular docking. So we have different definitions. In general, I'll say molecular docking in the sense how effectively a drug binds to the receptor. If I'm giving a drug, how it goes and binds to the receptor, how strongly it is binding. So that prediction is called as molecular docking. So docking is the identification of low energy binding modes, of small molecules or ligands within the active site of a macromolecule or receptor whose structure is known. And docking is the means in other terms, it's a computational determination of binding affinity between molecules, that is the protein structure and ligand. Or sometimes we will be working with the protein also. We'll be working with the protein also. So in other term, if I given a protein and ligand, they are going to find out the binding free energy of the complex formed by docking them. So this is how this thing, ligand with the receptor, how it goes and bind, what is the energy obtained, how efficiently it is getting binded. That's it. We are going to predict that. That is there to be a molecular docking. So why modeling is required? Experimental determination of structure is still a time-consuming and expensive process. To identify a particular protein structure, it will take time, too much of time. But we have the sequences. So with that sequences, we can able to model the protein structure. One thing. And second thing is number of known sequences are more than the number of known structures. So here we may have the DNA sequence, RNA sequence. What are the sequences we have? Gene sequences will be having. Based on the sequences, we can able to convert them as a structure. So number of sequences are more when compared to the known structures. That is the reason modeling is required. And then the most important thing is structural information is essential in understanding the function. To understand the particular protein nature, if it is a beta turn or alpha helix, what is the turn, how many sheets are there? Based on that, we can able to as I say what type of activity it will do, what type of reaction it will make. So for those things, modeling is required. So in a publications or somewhere else, you may have seen the word in silico. In silico is the expression used to why we are using in the sense. In general, pharmacology evaluation usually will be studying as a in vitro and in vivo. Rather than that, the new term is called as in silico. In vitro, outside the body; in vivo, inside the body. That is the general terms we will be using. The term in silico, where we'll be using in silico in the sense, the works, the pharmacology evaluation of a particular drug or moiety which have been carried out using the systems, using the laptops, computers, is said to be in silico. Why? Because the systems are interconnected, means within the boards, are made up of silicon. That's why it is said to be in silico studies. That is the reason. So in silico drug design is defined as the identification of drug target molecule by employing bioinformatics tools. Bioinformatics tools are many tools are available. Not only molecular docking and dynamics, or other than that, many tools are available. So in general, if an activity is predicted using the system, it will falls under the category of in silico studies, in silico pharmacology evaluation. So types of in silico drug design. So there are two types of in silico drug design is there. One is structure-based drug design, another one is ligand-based drug design. The word itself, it will say ligand-based. If a drug moiety is available, based on the drug moiety, if we are designing a new moiety, it will falls under ligand-based. And if it is a structure-based in the sense, if you know the protein structure, based on the protein structure, we'll be working out for finding a new drug. I'll explain in detail. So receptor based or structure based strategy. So this uses the 3D structure of a target receptor to search for a potential candidate compound that can modulate the target function. If you take a protein, or amino acid, that it will have an active packet. Based on that active packet, if you are trying to find out a new medicine, irrespective of the class of drug, like it may be belongs to pyridine category, pyrimidine category, oxazolone, whatever the thing may be, it will try to accommodate which is best. Then we can able to go. This involves molecular docking of each compound in chemical database into binding effector. That's what I said. Chemical database. The database will have lakhs and lakhs of molecules. So based on that, which is having best effect, best fitting effect, that we can able to take it into the account. So the compounds will be ranked according to the scoring function. That scoring function and docking, these things I'll explain in future slides. So scoring function such that the scores correlate with the binding effect. And then next comes the receptor based methods have successfully applied in many targets. That is general thing. So ligand based. If you doesn't have the structural features, if it doesn't have the structural feature of a particular protein, and moreover, if any sort of other drugs have been treated, used for the treatment, we can able to take the drug and we can able to work out for those things. That will fall under the category of ligand based strategy. And then moreover, structures similar to the known inhibitors are identified from the chemical databases by variety of methods that can be used. Example I'll give. If you take the ciprofloxacin and norfloxacin drugs, ciprofloxacin is the quinoline derivative. Same norfloxacin is also quinoline derivative. Both will have a more potential. So this will fall under the category of medicinal chemistry classification. So medicinal chemistry classification, how we will be classifying? Based on the nucleus structure. Based on the nucleus structure. So similar nucleus, if are available from the databases, if you are screening for that and if you are identifying from that, it will fall under the category of ligand-based strategy. So some of the methods widely used are similarity and substructure searching, pharmacophore matching, or 3D shape matching. That is substructure is related to the structure. Some functional group modification will be done, and then pharmacophore matching, making the pharmacophore model, which is the active means which functional group is responsible, whether the aromatic ring is required. That SAR studies we are studying now. Based on that, the pharmacophore matching has to be done, and 3D shape matching will fall under the same category. So numerous successful application of ligand-based methods have been reported. So basic binding mechanism. We are saying the mechanism of binding efficacy and those things and all. We have to know. We have to know what is the basic things behind the screen. What is that thing happening? So it is based on the complementaries between the ligand and binding site. It is based on the complementaries between the ligand and binding site. So first thing is stereocomplementaries. That is shape of the ligand is mirrored in the shape of the binding site. You might have studied the chirality and those isomers, those related, right? It will fall under such kind of things. The shape of the ligand should be mirrored to the binding site. And then next comes the physiochemical complementaries. So these two complementaries plays a major role in the binding efficacy, binding mechanism between a protein and ligand, between a protein and ligand. So types of docking. If you take, we are discussing about the docking and those things, and we have to know what are the types of docking. Two types of docking are available. One is rigid docking, another one is flexible docking. Rigid docking in the sense, in the enzyme class we have studied lock and key model. Lock and key model. For a particular lock, only a particular key will open. Right. The same way here, both the receptor and ligand will be kept as rigid as such. So how it will try to bind, how it is binding. If you are working on that, it will fall under the category of rigid docking. And flexible docking is induced fit docking. Here, if it is having a rotatable bonds, will be making them as a flexible one, and we'll be trying to find out how it is performed, how it is working out, how it is working. So based on that, we can able to calculate what is the best score, which type of force is giving better score. Those things can be predicted. And then categories of docking, that is types. Here the categories in the sense we can able to do based on two categories: protein docking as well as protein ligand docking. Protein docking in the sense here both the receptor and the ligand both the molecules are rigid. Interaction produces no conformations in the changes. Interaction produces no conformation in the changes. Here, both are proteins. Both are protein nature. And then here the protein ligand docking is ligand is flexible, but the receptor protein is rigid in nature. So here the interaction produces conformational changes in the ligand. I'll show the image. So protein docking. So computational modeling of quaternary structure of complex formed by two or more interacting biological macromolecules. So when we'll be using this in the sense like you have to find out if the protein structure is not available for a particular thing, alpha 2 beta 3 like that. If the naming is at nicotinic receptor is there, alpha 4 beta 3 like that. If the changes, we can able to carry out the protein docking and to make like in place of homology modeling. So where it get interacted, based on that interaction, we can able to make the chain and we can proceed further. This is one way. And then the next way is protein complexes are most commonly attempted targets for modeling, followed by protein nucleic acid complexes. For these things, it is used. And then next is a protein ligand docking. This is a widely used method. Widely used. Around 90% of been 80% of the people are working on protein ligand docking only. Is to predict the position and orientation of a ligand molecule. So here the protein structure is there, ligand structure is there. How it got binded from the complex, we can able to see the 3D structure, how it got interacted, and how it got bound to the protein, or how it got bound to the receptor. That will fall under the category of protein ligand docking. So we are using the term called as docking and scoring. So to place a ligand, that is small molecule, into the binding site of a receptor in the manner appropriate for the optimal interaction with the receptor. That is it. Here, docking in the sense ligand will be there, receptor will be there. This receptor ligand will try to get modified in the sense it will try to bind with the cavity in different poses. You can be able to see the change in the shape. You can be able to change the shape. This is said to be docking, trying to interact, trying to bind. So what is the amount of energy? What is the energy found? That is said to be scoring. To evaluate the ligand receptor interaction in the way how it get experimentally absorbed mode from the others. So what is the binding affinity? If you are calculating the binding affinity, that binding affinity will fall under the category of scoring. So what are the components of docking? So there are two components. The docking will fall under the search algorithm. Typically, protein ligand docking software consists of two main components which work together. If you take any software for doing the docking studies, it will work on two mechanisms. Not two mechanisms, two main components. One is the search algorithm, another one is scoring function. So search algorithm, what it will do? It will generate a large number of poses of a molecule in the binding site. So here I have showed different poses it's trying to bind it. It's trying to bind it. So based on that, it's working out one thing. And then scoring function. The scoring function is the score. Calculates the score or binding affinity for a particular pose. What is the score? What is the binding efficacy of this particular pose? What is the score for this particular pose? So it will calculate. So the binding affinity or scoring representing the strength of the binding. So how based on the score we can able to say how it got interacted, how it got bounded strongly. So the scoring function will take place. What are the major parameters? It will calculate the hydrogen bond interaction between a protein and ligand. It will calculate the electrostatics energy between the protein and ligand, van der Waals force, hydrophobic interaction, pipe interaction, etc. So whatever the forces are there, it will calculate each and every forces, and it will make out, and it will give the calculation. The energy will be out. The energy will be in the negative. The energy will the energy score. What are the most of the softwares will give in minus only. Negative higher is said to be more potent in binding, more efficacy in binding. If example I have 3 compounds which I have done docking for a particular receptor, I got a score one. Let it be A, B, C. For A, I got a minus 6.5. For B, minus 7.5. For C, minus 6.8. Out of these, minus 7.5, B is the topmost, which is having a best binding efficacy. So negative higher is to be more stronger in interacting, more stronger in binding to the drug nature. So applications of molecular docking in modern drug discovery, how it is get used. So it is helpful in determining the lowest free energy structures of the receptor ligand complex. And then next comes the search database and rank hits for lead generation. And then comes the calculate the differential binding of a ligand of two different macromolecular receptors. If I take a, because people may have the secondary effect or adverse effect or secondary pharmacological effect will be there. So to a particular receptor where we have targeted, other than that target, whether our drug goes and bind to any other target. For that we can able to predict one thing. Second thing, we have said the complexity of the drug discovery, selective in nature. It should be selective in nature because we are having adrenoreceptor, alpha 1, beta 2, alpha, beta, like that. We have this thing to find out their efficacy. We can able to use this drug discovery molecular docking, and then study the geometry of particular complex. What is the how it get binded? That is lock and key method. Propose modification of a lead molecules to optimize the potency or other properties like DDO drug design for lead generation. So here the lead molecules from the drug discovery process, I have thousands of molecules and it's a new disease or there is no other drug for a particular disease. What we can able to do, we can able to choose that ligand, choose that protein. We can able to do the molecular docking, that is virtual high throughput screening with the thousands or 10,000 molecules. We can able to find out which type of nucleus is working. From that, we can able to make them as a lead moiety. We can go for further. And for library design, screening for a side effect that can be caused by the interaction with other proteins like protease, cytochrome P450, and this also can be done. These also can be utilized. So docking is also widely used tool in predict protein-protein interaction. Also, it is used. Now, proteomics, not proteomics, peptidomimics, peptide synthesis. These are involved in the drug discovery process because of their more efficacy, strong efficacy. That's why they have been utilized. For that prediction. While designing a protein essence a peptide, how it will work out. Further, we can able to use this docking studies. So knowledge of molecular association aids in understanding a variety of pathways taking place in the living and in revealing of possible pharmacological targets. What are the targets? Like using the proteomics and genomics, we can able to do. And then docking by virtual thread high throughput screening, and this is less expensive when compared to high throughput screening, faster than conventional screening. We can able to scan, means we can able to evaluate a number of lakhs and lakhs of molecule within 1 or 2 days using the computational support in a less time. So high throughput itself a trial and error approach, but can be better complemented by virtual screening. That also can be done. So here are some examples, some proofs which have been proved by the applications of molecular modeling studies, molecular docking studies, computational drug design. The drug available was dorozolamide, which is an carbonic anhydrase, sequinavir, HIV protease, reliance and neuraminidase, and discovery of indinavir, that is a HIV protease inhibitor, identification of haloperidol as a lead component in structure-based design for non-peptide inhibitors of HIV. So these are some ideologies, some proofs. In the drug discovery process. So by seeing this, or you may have a plan to do a research work to carry out the molecular modeling, to carry out the molecular docking studies. What are the things required? So the prerequisites for molecular docking, a valid hypothesis should be there. A valid hypothesis should be there. Without hypothesis, you should not do the work. I can give an example. In olden days, if you are not feeling well or something else happened, our grandmas and grandpas, they will take up medicine that is of natural source, natural ingredient. They'll make a kashaya, they'll squeeze out the juice from that and they'll give. But for those things, we doesn't have a proper proof. So with that hypothesis, which will be written in medicinal plant books or something else. I'm just giving an example. So taking that into consideration, you can able to work out. And then for that, we require optimized protein structure. So which disease you are going to work out? For that disease, you have to study the complete pathophysiology and reported papers, and you can able to find which receptor, which enzyme is playing as a culprit, which receptor or which enzyme is playing as a culprit. We can able to know them. After knowing them, we can able to download those structures from the protein data bank. Protein data bank. There is a data bank available in the website. It is a free RSC PDB website. So that data bank will have a protein structures. From that protein structures, you can able to download and you can able to optimize the structure. One thing. And second thing is optimize ligand structure. As we have the hypothesis, some curcumin is there. We'll take a curcumin if you are taking. It can be used for the antibacterial. That can be used. So taking that into the consideration, we can able to go further process. Go to the further process. We can able to utilize those things. The ligand structures can be downloaded from the PubChem. PubChem database is there. Zinc database is there. ChemSpider is there. N number of databases are there. I'll show those databases next slide. So that can be downloaded, and you can able to optimize it. Then for molecular docking, softwares available. List of softwares available in the sense through web server. Web server in the sense they will have their own source through online. We can able to upload the protein structure and ligand structure. We can work out. And freeware tools in the sense which are available for free to use, download and use. Academic freeware tools. Certain softwares, what they will do, they will pay, they will give free only for academics as a like a evaluation version, trial version. And then commercial tools are available. So freeware tools most probably widely used was Autodoc was used. Academic freeware tools, some are there. I'm not sure with those things because and commercial tools. If it takes Scottinger's Glide is there, Gold is there, MOE is there, Discovery Studio is there. They will be giving us a trial versions for 1 month or 15 days. You can utilize those things also. And then visualizing tools, academic freeware tools. CSAR is there. Zastar they are giving only for academic purposes. Not you can't able to perform the research or publish the paper. You can able to show us a demo for the students. And then the visualizing tools, tools like a PyMol is there, and then online also PLIP is there. So these are the basic requirement for carrying out the molecular docking studies. So here some databases which are given. So impact. So databases is based on the marketed drugs, published papers, and other things and all. So there are some databases. First for 4 databases, that is impact, AromaDB, Dr. Duke's phytochemical and ethnobotanical databases, MAVP. So this will have the data. These are the databases most widely for the plant sources. What are the active constants isolated from the plant sources? For those things, these 4 databases can be utilized. Then it comes the Chem database, Zinc database, Zinc 15 database, Chembel is there. So these are the databases for the synthetic drugs. And then ChemSpider, Drug Bank, PubChem, TCM, CHEPA, chemical entries for biological interest. So these are the databases where based on the different countries, different peoples are utilizing and keeping those their sources. So these are the databases where you can able to download these structures and you can able to work out. And next comes the protein, where you can able to find out the targets. Protein data bank, binding MOAD, that is mother of all database. That name itself binding MOAD. So from this database, PDB bind NR list, that is nuclear receptors, ligands, structures benchmarking database. So these are the databases where you can able to identify or download the protein structures. Most widely used database was protein data bank for finding the protein structure. This will have an alphanumeric in nature. This will have the alphanumeric on nature. The drug structure will be alphanumeric, 4-digit only. It will contain alphabet as well as the numbers. That is the thing. So here I come to the conclusion part. Molecular docking will give the promising effect on identification and optimization in modern drug discovery. The combination of chemical information of a natural products with the docking-based ligand virtual screening will play an important role in drug discovery in the post-genomic era, as more and more new potential targets are emerging from the functional genomic studies. And then docking-based virtual screening lead to such higher hit rate than the traditional screening, that is high throughput screening method. So docking method provides an opportunity for designing a drug or designing of active compounds. However, it has been emphasized that docking-based virtual screening is not the replacement of actual experimental screening. Here you have to keep in mind, only with the docking score, you can't able to say the drug will be potent. Here the drug will say whether it is having binding efficacy or not. Theoretical calculation only. In biological, if it's going, it may be 50-50 only. The reason is the drug, once it is administered, it has to cross all the biological barriers. So once it reaches the target, then only we can able to say the efficacy. So docking will give whether it will bind or not. That's all. As a matter of fact, these two methods are highly complementary. Without high throughput screening or computational screening, it will be helpful for each other. So if you have any other questions, I'm happy to answer the questions.
Thank you, sir, for the wonderful talk. Thank you for enlightening us with the vast applications of molecular modeling in drug design. Your explanation has enabled us to have a better understanding on computational chemistry, in silico drug design, types, categories, components, applications of molecular docking. Thank you for the talk, sir.
Thank you.
Now the session is open. Thank you. Now the session is open for questions and discussions. I invite Professor Dr. S. Jayaprakash to coordinate the discussion session.
Thank you, madam. Thank you, sir. Thank you for your valuable lectures. And I'm requesting all the participants, please raise your hand for a question session. So anyone having the questions, please raise your, will unmute you. You can directly ask that questions.
Sir, Jayaprakash sir, some have asked the question in the chat box. Shall I
Yes,
answer?
sir, yes, sir.
Reading the
Yes,
chat.
sir. Yes, sir.
So I couldn't able to spell his name. Muragad. Sir, does this protein data bank is free? Yes, absolutely it is free. You can able to download from the directly. If you just search protein data bank or PDB data bank in the website, you will get the clear idea. Means that the first link will be protein data bank only. It will be RSC PDB. RSC PDB. That is the thing. Is there any specific protocol to be followed for protein ligand binding? Yes, you have to follow the proper protocol. As I showed, right? Protein has to be optimized, ligand has to be optimized, those things to be done.
Sir, one more question from my side. The plant phytochemicals, so which are active in that molecular modeling, so that output can be patentable, sir. So what are the phytochemicals? Hello? Sir? Hello?
Oh, sorry, you have muted me.
The plant phytochemicals.
Yes,
So
sir. Yes,
whatever
sir. We
showing
can,
activity.
sir. We can able to do, but not only with the in silico studies. You can't able to do the pattern. You have to isolate the particular compound. You have to carry out the pharmacological study, and then you can able to prove for that molecule to the pharmacological activity. You can do, but not with the in silico studies. It can't be patentable.
So is there any specific protocols for doing each software, sir? So it's any specific site or books available for carry out the molecular studies, sir?
Research papers and YouTube videos and many are available, and moreover, all the software they have given in their working protocol in their software tools only, sir. Example,
Okay,
most
sir.
probably what I will suggest is you can go with the Autodoc or Autodoc Bina only. Those two are reliable source for, because many colleges can afford the payment. Many can't able to pay and get the software. During that time, what they can do, they can utilize this freeware which is worth. If you take Autodoc or Autodoc Bina, it is provided by the Scripus University in I don't know the country name. Scripus Institute. They are providing. It's a wonderful software. You can utilize those things.
Okay,
If
what
anyone want
about
anyone want the help, they can ping me. I'm ready to help because I'm working on that only.
okay, sir. Thank you, sir. So another one, our participant like to ask one question, sir. Prajakta. Madam, please unmute yourself and ask your question.
Yes, sir. Share the PPTs and link of this today's webinar. Hello,
Yes, ma'am. Yes, ma'am.
sir.
They'll share you, ma'am.
Yeah,
We'll share. We'll share. We'll share.
yeah.
Hello, sir. Sir, can I
Yes.
speak one?
Yes.
Thank
Yes.
you.
Yes.
Manish.
Yes, sir. Thank you for the session, sir. It's very highly streamlined, clear, informative. And sir, I want to ask one question.
Yes, sir.
Actually,
Please.
I'm a PharmD student. I'm in my internship. I want to pursue my career in drug discovery, like docking and computer drug design.
Yes.
Can I please know how to enter drug discovery from PharmD?
Yes, child. I can give you one suggestion. One of my junior, she have done her PharmD and now University of London. She is doing her MS in drug discovery process. She have completed that also. It is easy to means it is not that much difficult. You can able to enter. The thing required is you have to be anyway as a PharmD. You are saying you will be strong in pharmacological process. That is the pathophysiology. You will be strong. The disease condition will be strong. So based on
Yes,
that,
sir.
you can able to enter. You can able to help the people in gene profiling, gene expression studies, those things and all. Because drug discovery using the computational tools, it's a vast region, it's a vast area. So we can able to make more and more. You can enter more and more regions. So if you want further help, I can help you. Just mail me. I'll share her detail. So from her, you can able to get more guidance also.
Yes, sir. Sure, sir. Thank you very much, sir. The session is very excellent and it's highly streamlined, actually. Thank you, sir.
Thank you. Thank you. Thank you.
Thank you, sir.
Hello, sir.
Yes, ma'am.
Hello. Yes, sir. I am from Pune, PhD student.
Yes, ma'am.
Just I want to ask my question. With my previous question, I have asked you that whether there is a protocol. You have said yes, it is a protocol that you have to optimize the lead and the ligand also. So
Yes,
I
ma'am.
think from my literature study, I have taken some papers where already the cell line study was there conducted, and from that I have taken the specific protein. Okay, they have already optimized it.
Okay.
But only with the cell line studies. Now I have my chemical this ligand also, which is also optimized. Okay, just I want to repurpose that product, that drug. Okay, already it is in the market, but I want to repurpose it. So for now, I want to do the docking study. So what I want to ask is that whether now the
Yeah. I understand. I got your question. The thing is, I said optimization of ligand and protein, right? That has to be done while doing the docking study. There is a protocol. I'm not saying about the what are the things you said, right? Already the cell line have been done, those things and all. That will comes under the valid hypothesis part. It will comes under valid hypothesis part. Now, what I am saying is here. I'll take this slide. So here you can able to see the terms optimize the protein structure and optimize ligand structure. While taking from the protein data bank, if you're downloading, it may have some other substitution other than the protein. It will be containing the co-crystallized structures also will be there. That you have to remove and you have to optimize that structure. That I said. You can able to work out using the tools like Swiss PDB viewer is there. Swiss PDB viewer. If you are opening your protein molecule into that, we can able to see what are the constant present in that. From that, we can able to purify that protein and we can able to do the further studies. Optimization of ligand structure in the sense for doing a particular docking studies, they may have software to software it will differ. In the sense, what type of input it require. Because they are mathematical calculation. we have given in certain format only. Certain time they will ask only smileys. They will ask SDF file format. They will ask us CDX format. So for that I said the optimization.
And then after that optimization,
We can
is
able
there
to
any?
carry out the docking studies, ma'am. We have to carry out the docking studies using the work protocol given for the particular software. Whichever software you are going to use, for that definitely there will be a working protocol. You can use those things. In case if you need further help, you can mail me.
One more question is there. You said that when that ligand and protein binding, if that you can this energy is in the negative form, okay,
Yes.
then it is the maximum one.
Yes.
So is there? I mean, what is the or you can say the
It is based
what
on
is the
the, ma'am,
reason behind this?
yeah, it is based on the wave equation. It is based on the wave equation, Schrödinger's equation or wave equation. That equation formula is like that only. It is pre-built. It is inbuilt into the software. Whatever the interaction between the protein and ligand, that is hydrogen bond interaction, what is the value? It will be placed into that particular space. And then for hydrophobic interaction, van der Waals force, what are those things? It is based on that only. It is based on the wave equation. Wave equation will give in the negative way only. That is the reason for that.
Thank you, sir. Excellent session. Thank you.
Thank you. Sir, another one more question is,
Yes, sir. Yes, sir. Happy
so
to
if
answer.
that docking is any molecule, it is not obeying the Lipinski rule,
Ah.
whether it is can be further can proceed for docking
Yes.
molecular modeling,
Yes,
or
yes.
any other options are there?
Yes, sir. We can able to utilize. Example, I'll give. If you take a drug of TB, rifampicin molecular weight is more than 2000, but they are using, right? It can be, sir. Certain times we can able to violate the rules.
So
It
is there any rule like a protocol like these are the rules can be violated?
No. It's not like that. Sir, it is sir. Lipinski rule is made. If it is within this, it will be good. He has suggested. It is not a particular proper rule. You have to obey. It is a suggestion. According to me, what I read, what I felt was I am saying. So according to me, it is a rule. It's a hypothesis. What he have done, he have taken some 1,500 molecules which have been marketed. From that, he have taken and he has suggested. So, if a molecule is within the molecular weight should not be more than 500. Number of donors should be not more than 10. Acceptor not should be 5. That log p value should not be 5. So, they have given in that basis. If it is within this limit, it will be good, like that they have said. Nowadays, many drugs have been marketed with violating their rules, sir. We can, sir.
Thank you, sir. Thank you very much. Sir, another one more question from audience. So,
Yes, yes.
what are the software available for binding site prediction?
Sir, finding the
Binding site prediction.
binding site prediction, that is active site prediction.
Yes, sir.
We can able to use active site predictor. CAST-P is there. CAST, CAST-P is there. Active site predictor is there. And moreover, if you are downloading from the co-crystallized structure, if example silica will be binded to the COX-2 inhibitor. That COX-2 protein will be available in the protein data bank. You can download and find the active site of the particular standard drug. So that what are the active sites it have binded. With that, if you are trying to bind with our drug structure, it will be much more efficacy. Much more, it will be better. CAST-P is available, sir. CAST, CAST-P.
So, freeware, sir?
Yes, sir. Yes, sir. Online it's freeware. CAST-P, active site predictor, finder.
Sir, another question.
Yes,
if
sir.
you are changing the grid coordination, so the
Come again, sir. I couldn't get it.
Grid.
Okay.
Grid coordination, if you are changing, so it will give a reproducible result, sir.
And moreover, I'll say, even if you say if you keep the proper grid box, that is 90, 90, 90 if you are keeping, and if you are trying to reproduce the result, we won't get the proper result back to back. And the in vitro studies, what we are carrying, in vitro studies, we may get the reproducible result. But in computational tools and softwares, we will never get back the reproducible results, because it's a wave equation and it is happening in the nature. So we can't able to find out. Means we can't be able to get the reproducible results.
Sir, my personal question. So, is
Yes,
there
sir,
any
yes, sir.
server molecular dynamic studies, server based?
No, sir, no, sir. No servers are available. Very rare, sir. Very rare in the sense within their university they are utilizing. They are not giving for the peoples.
So what is the minimum requirement, sir, for running the molecular diagnostics?
A GPU, sir. Minimum requirement of GPU, NVIDIA GPU, sir. At least 1050 TX, 1050 Ti TX is the minimum quantity. I'm saying it will have the CUDA core of 800 something, because I have that recently 2-3 months back. Not 2-3 months, like 1 year back I got that. So I'm replacing that only using iFi system. Core iFi system I'm using with that, and I have increased the RAM size to 10 GB. So with that I'm running, sir. At least a graphics card of NVIDIA CUDA GTX 1050, sir.
Sir, another question from audience side, the participant side.
Yes, sir.
A protein contain 2-3 co-crystal. How can we choose the specific co-crystal out of this for our ligand interactions?
If a protein contain 2 to 3 co-crystal, how can it is not possible? I don't think so. 2 to 3 co-crystals will be present for one drug structure.
My personal observation also, I just found some structure with 2 different ligand binding crystals.
I don't think, I have not tried, because why I'm saying in the sense active site, active pocket of a protein. According to my experience, I'm saying according to the protein structure, it will have an active site. If already one co-crystal is went and bind, it's simple, sir. Only one chair is there. You are sitting. If I want to sit now, I have to take you out and then only I can sit. That is the thing. So 2 to 3 co-crystals in the sense like already binded on, again they will try to bind with the other binding sites. They will try it for the other binding site. For an active site, it is this 2 to 3 co-crystal when they will use, maybe I'm not sure, I have not crossed again, maybe only when they are trying to find the dual binding efficacy, or else like for the same disease they will try to give 2 drugs, right? In such kind of condition they might try it. Only based on the active site they can able to do so.
Okay, thank you, sir. Thank you very much, sir. So still there are a few more questions, sir. Due to that time, just we will post that question, sir.
Fine, sir. Fine, sir. You can, sir. You can ask any number of questions.
So, okay, sir. I'll hand over this session to the Preeti madam for the. Thank you, sir.
Thank you, sir. Thank you, sir.
Madam, Preeti madam.
Thank you, sir.
Thank you, ma'am.
As a token of our appreciation and gratitude, we would like to present the virtual memento to our speaker, Dr. Parasuraman. Inviting Principal Dr. V. Alagarsamy, sir, to present the certificate and memento to the speaker and conclude the session with the vote of thanks.
Good afternoon, all the participants of this wonderful speech by Dr. Parasuraman. I think Dr. Parasuraman delivered the lecture in a simplified manner, because many of the researchers or the students who are at the start of the research, they feel drug design is a very complicated thing that cannot be afforded or that cannot be learned by that is starters. But he has given the speech in a very simplified manner that makes everyone to think of to work on drug design and molecular modeling. So really the speech was very much informative and provided in a simplified manner. So I appreciate Dr. Parasuraman for his valuable time spent with us for the service of the students and the young faculty members. I also thank our management for giving this platform to share this knowledge with the worldwide. I thank all the organizing committee members of our MNR Educational College of Pharmacy. And last but not least, the speech what he has given and the clarification what he has given by Mr. Parasuraman, really it was very much inspiring. I hope all the audience must have used this chance, and really in the coming days also, even with the more practical oriented things also, we can get it from Dr. Parasuraman, because he is working in the same field for many years and he had got a good experience of on the drug design and molecular modeling. Thank you, Dr. Parasuraman, for your valuable time spent with us during this more than one and a half hour. So I, on behalf of MNR College of Pharmacy, I provide this certificate of appreciation for your valuable time. Kindly accept this certificate of appreciation, and definitely a long way to go, and we'll have many more such webinars with the participants. Thank you one and all. So I request Dr. Parasuraman to accept this certificate.
Thank you, sir. Thank you so much for the opportunity, sir.
Okay, sir. Thank you one and all. So I think Dr. Saravanan, we can sign off.
I would like to give the acknowledgement to my institution, Rama Institute of Applied Sciences, and most probably to the all the participants who are patiently listening and they have raised their questions, and most probably the person who have given me opportunity to Professor V. Alagarsamy sir for the opportunity, and their organizing team members, Saravanan sir, Jayaprakash sir, and one more sir. I couldn't able to remember his name. Sorry, sir. So on behalf of thank you so much, sir.
His name is Bose, sir.
Bose sir. Sorry, sir. Bose sir. Thank you so much for the opportunity, sir.
Thank you, Nagar.
Thank you.
Thank you, one and all. So I'm requesting all the participants to submit your feedback, and once you completed that filling in a feedback form, you will receive your certificate within 3 days to your registered email ID. So that feedback link, many people they got messaged that it was not activated. Once the completion of the webinar only we will activate. So now it was active. You can submit your feedback. Okay, we will meet you in that another webinar with a good topic. Thank you, one and all.